Welcome back, everyone. So we'll finish off the first topic. We're going to talk about cellular adaptations, and these are various cellular adaptations that can happen depending on different scenarios. Some of these you've probably heard about before. In fact, most of them you probably have. When we talk about cellular adaptations, they're just changes that cells undergo as they adapt to various environmental changes. Now, the type of change that can actually happen within cells, you can have an increase in number of cells or a decrease in number of cells. You can have an increase or decrease in the size of the cell, but you can also have a change in the type of cell that's actually present within a tissue. And all of the changes that occur within the cells, it's not limited to just one. You can actually have multiple changes at the same time. You can have an increase in number and size of the cells. You can have an increase in number and type of cells. It's not just one thing that has to happen. So the first cellular adaptation that we'll talk about is something called atrophy. And atrophy is essentially a decrease in the cellular size. A lot of times, atrophy is also referring to a decrease in the number and size of the organelles that are within each cell. So if we're looking at normal cells and we see the atrophied cells, these cells are smaller. And if you looked at their organelles, they would also be smaller and there would be less of them. There's lots of reasons why atrophy occurs. Atrophy can be physiologic, or it could also be pathologic. So an example of, so physiologic would mean it's part of the normal process within the body. Atrophy does occur normally. If you think about a lady that undergoes menopause, if you were to actually look at her uterine lining, the endometrial lining would have less glands after menopause. And that's related to a decrease in the amount of hormones that are released. And it's no longer able to maintain the proper amount of glandular cells within the endometrial lining. That would be physiologic. That's a normal process that happens. Oftentimes, atrophy is also pathologic. So for example, you can have something referred to as a disuse atrophy. If you don't use it, you lose it. So muscle atrophy. Imagine that somebody has, I don't know, they've broken a bone and they have a cast on. Over the next 6 weeks, they do not use their limb where the cast is on. Those muscles are going to atrophy. They're going to get smaller. And it's related to the fact that it's not being used. So it's a disuse atrophy. You can also have atrophy related to denervation. That would be related to there being no nerve input to a particular part of the body. Oftentimes that's a muscle. Without nerve input, the muscle can't work. And without the muscle working, you can't actually have any, you can't maintain the cellular size. The loss of endocrine function, that would be reduced hormones to maintain the glandular epithelium of the uterine lining. You can also have a decrease in blood flow or malnutrition that can actually lead to atrophy. No nutrients going to the cells, they can't be maintained. Here's an example of an atrophied endometrium. This is on the left, you see a normal endometrium in a lady of reproductive age. You can see all of these circular oval areas. These are all endometrial glands. And these endometrial glands are numerous. If we look at the same biopsy in a lady after menopause, you'll notice that there are very few endometrial glands. The cells have decreased in number, so they've atrophied. They're also smaller. If there are glands, they are smaller. They're not as well maintained. So this would be an atrophied endometrium that would happen after menopause. Hypertrophy is the is really the opposite. It's an increase in the cell size. And this is related to an increased stress on a particular body part. In general, we see hypertrophy in tissues that can't adapt to an increased workload by increasing mitosis or forming new cells. Instead, these cells can just get bigger. A good example of this would be muscle. Muscle can't undergo mitosis. So instead, they just get bigger. What we're looking at here is an example of a hypertrophied myocardium. So the heart muscle, this would be the atrium, and then here's the ventricle, probably the left ventricle. Very small chamber size, very thick myocardium. In this case, this would be a pathologic hypertrophy. If you were to look at these cells under the microscope, they'd be much bigger. They've had more muscle fibers within them. But we do see changes within the myocardium depending on what factors somebody is exposed to. It could be physiologic. Well-conditioned athletes we know get left ventricular hypertrophy. So the left ventricle muscle wall increases, those cells increase in size. That doesn't change cardiac function at all. The pathologic causes would be if there's too much pressure within the left ventricle or too much volume. We're going to talk about this later, and we're going to talk about what the overall effects of either a pressure overload or a volume overload is on the heart and the rest of the body. You can have hyperplasia. This is an increase in the number of the cells. In general, we see this in individual or in tissues that can undergo mitosis. So the epidermis, we see it, intestinal epithelium, most glandular epithelium, we also see it. In a very good example of hyperplasia would be callus formation on the skin. So somebody lifts really heavy weights, they don't wear gloves. Over time, people will develop calluses on their palms because of the heavy lifting. That increased stress placed on the epidermis is going to cause those epidermal cells to undergo hyperplasia, to get thicker to prevent injury. Or a guitar player getting callus formation on their fingertips. Also uterine and breast enlargement during pregnancy. This is an example of hyperplasia. The glandular tissue is going to increase in number, and that's going to be an example of the cells dividing and becoming hyperplastic. These last two, first one is metaplasia. Metaplasia is when one adult cell type is replaced by another adult cell type. Almost always metaplasia is related to chronic irritation and inflammation in a particular body part. Because one cell type is turning into another cell type, another normal cell type, but in the wrong place in the body, this is often indicating that there's some sort of a there's some sort of a change in the genetic material of the cells, which may indicate that there's a mutation, which may predispose individuals to cancer. Metaplasia tends to be reversible if you remove the irritation or the inflammatory component. Good example of it is this. What we're looking at here is a section through trachea. So this is the mucosal lining of the trachea. Then what we see here, this bluish area is the cartilage. These are chondrocytes with cartilage. And then we have the muscular layers underneath. Normally what the trachea is comprised of, the epithelial lining of the trachea. You can see these little hair-like projections. This is ciliated. These are ciliated cells under normal circumstances. And it looks like there's many layers of cells. There's actually not. These little blue dots, these are all the nuclei, and they're just scattered throughout the cell. Each cell has a nucleus in a different spot. So it looks like there's multiple layers, and it almost looks like there's strata of these layers are in strata, but they're not. So we call it pseudostratified. So this is ciliated pseudostratified columnar epithelium, really tall cells, taller than they are wide. This is normal for trachea. In an individual that smokes, that chronic irritation to the lining of the trachea causes these pseudostratified ciliated columnar cells to change into really flat cells. There's no cilia left. And these flat cells are squamous epithelium. And this is associated with cigarette smoke. So changes to squamous epithelium. Squamous epithelium is normal. It's just not normal in the trachea. So that's an example of metaplasia. We'll talk about another example of metaplasia in our GI section. Dysplasia is the last cellular adaptation we'll talk about in this section. It's really deranged cell growth. Cells are going to vary in size, in shape, in the organizational structure. It's also related to irritation and inflammation. And while it's a strong precursor for cancer, it doesn't always lead to cancer. Oftentimes, if the irritation and the inflammation is removed, the cells can return to their normal structure and function. A good example of where we look for dysplasia is in the uterine cervix. And this is with Pap tests. We look for changes within the cells. So what we're looking at here on the left side is a Pap smear where they took some of the cells of the cervix. These are normal squamous cells of the cervix. Notice that there's the entire cell is this sort of lighter shaded area. That dot in the center, that's the nucleus, and there's tons of cytoplasm in there. That's a normal squamous cell of the cervix. Depending on the irritating factors that the cervix is exposed to, usually this is in the form of a particular infection called human papillomavirus, or HPV, can lead to changes within the cervix. And eventually those changes can lead to cancer. This is one of the reasons that we screen for cervical cancer by doing Pap smears. These changes, it doesn't go from normal to cancer right away. There's grades of abnormal cells. There's this, you don't need to know what these stand for, but the L stands for low grade, the H stands for high grade. And the low grade would be slight changes within the cells, but you can still sort of recognize them. Notice that here's a squamous cell. There's sort of a strange nucleus there. You might see a nucleolus there, but it's still, it looks a little bit like the normal. In the high grade, as the changes get worse, you can see that these cells have a huge nucleus, huge nuclei, and very little cytoplasm. This is very abnormal compared to the normal squamous cell. And if this progresses, sometimes this dysplasia, this would be considered dysplasia. This is also dysplasia. This dysplasia can progress to cancer. This is almost not even recognizable to the normal squamous cells. And there's a couple of clues that this is cancer. Number one, it's very difficult to tell what tissue this came from. So sometimes that is an indicator of cancer, but not always. You can also see that there's some really strange looking nuclei with really prominent nucleoli within it. So here's a dark staining nucleus with really prominent nucleoli in the center. You can also see this little thing here, this really dark staining structure. And this is a cell that is in active mitosis. Active mitosis, the chromosomes are splitting apart from each other. It's very rare to capture a cell in active mitosis when you do a biopsy. You might get one now and again, but it's really rare, and it's a strong indicator that these cells are dividing a lot faster than they should be. And that's one of the hallmark features of cancer. It's this unregulated cellular growth. So this is an example of where normal can become dysplastic because of an irritating factor like HPV, and that dysplasia could result in cancer, cervical cancer. The point is to try to catch the dysplasia in the stages before it turns to cancer, or if it does turn to cancer, to get it very, very early. That's the goal with PAPs. So that would be a secondary prevention strategy, PAP tests. There's already the changes, but they're asymptomatic. We'll talk a little bit about cellular injury and death. There's lots of mechanisms by which cells can be injured. A lot of times cellular injury is reversible, but the reversibility really does depend on how severe the insult is. It depends on how well the cell is able to deal with the injury. And most of the time, that's based on two things. Number one, the nutritional status of the cell. Number two, the ability of the cell to regenerate the injured cells. There's not a lot of cells in our body that can actually regenerate, other than like skin. There aren't a lot of cells that can regenerate properly. Many mechanisms of cell injury, one of them is physical. There's radiation injuries, there's chemical injuries, biologic injuries, and nutritional imbalances. We're only going to focus on a few of them, but we'll give examples of all of them. So the physical injuries would be some sort of, could be a mechanical impact with another object. You're hammering a nail into a wall and you accidentally hit your thumb. That would be a physical injury to your thumb, mechanical impact with another object. But physical agents don't necessarily mean mechanical impact. It could be an extreme of temperature, burns, frostbite. All of these extremes in temperatures are going to disrupt the cell. It's going to disrupt the ability of the cell membrane to do its job. It's going to disrupt the blood flow to a particular organ. It's going to alter the enzymes, the enzymatic activity within the cells. So mechanical impact is one, extremes of temperatures is another, electrical injuries is another. Usually this is the injury is actually caused by heat production from the electricity, but we'll actually look at a couple of examples of this actually right now. Electrical injuries of the skin. This is an example of electrocution. You can actually see that the skin is burned and that some of the skin has sloughed off from the burn. That would be an injury. That's a physical injury to the surface of this person's hand, the dorsum of their hand and over their thumb. The right-sided image is actually just a very interesting image. This is something that you'll probably never see. It almost looks like a fern pattern on this person's arm. This is actually somebody who was struck by lightning. These are called Lichtenberg figures. And the electricity, as it flows through the body from the lightning strike, it travels along really superficial blood vessels, and it causes them to almost become scorched. And it will create this sort of fern-like pattern on the skin. Radiation injury is based on the electromagnetic spectrum. And so the electromagnetic waves, they go from really long waves to extremely short waves. Really long waves are radio waves. The longer the wave, the less the energy that it has. So it's the really tiny waves that are the most likely to injure our cells, because those have such high energy that they have the ability to knock electrons off of atoms and molecules and to cause damage to our genes. So this is something called ionizing radiation. So that's on this left side of the image, ionizing radiation. That would include X-rays and gamma rays. It's anything above the ultraviolet spectrum, cosmic rays, cosmic ray photons. These are going to knock electrons off of atoms and molecules and create really reactive substances that will bombard themselves, and sometimes with our DNA, and it'll lead to mutations. This is one of the reasons that we protect ourselves against X-rays. So when you have a dental X-ray, they will cover the rest of you in a lead suit so that you don't have the rest of your body exposed to that X-ray. There is non-ionizing radiation. The non-ionizing radiation is anything that is below the visible spectrum. So infrared. This can still burn. Infrared can still burn. Microwaves can still burn, but it tends not to ionize. It tends not to knock electrons off of atoms and molecules. The UV spectrum is somewhere between the ionizing radiation and the visible light spectrum. This is what we get from the sun. And this is actually the number one risk for skin cancer. So if you're exposed to the ultraviolet light over long periods of time, if you get sunburns, that increases your risk of skin cancer because the skin cells are the ones that are getting all of that ultraviolet radiation. It's also damaging to your eyes. It increases risk of cataracts and something called macular degeneration. So it's really common for people to have ultraviolet damage, UV light damage to their eyes that can lead to blindness, could lead to blindness. So always wear your sunscreen, always wear your sunglasses. But those are examples of radiation injury. There's lots of other types of injuries we can have as well. There's chemical injuries, so medications drugs that are taken can injure the body. Different minerals, so even supplements can cause injuries. Metals, lead, mercury. These are all things that can accumulate in the body. Even gases can be dangerous. Like if you think about carbon monoxide, that's going to be an injury. That's going to lead to injuries. You can have biologic cellular injuries, so viruses, bacteria, something called a prion, some types of parasites. These are all things that are going to damage the cells, but are living organisms that are damaging the cells. We're going to talk about those in our mechanisms of infectious disease. And then various nutritional imbalances, either vitamin deficiencies or mineral deficiencies, or even the toxicities of them. It's possible to have too much of the vitamins and minerals. So we'll talk a little bit about how cells get injured. There's three basic outcomes. When a cell gets injured some way, it can either be a reversible injury, and then a cell will recover and return to normal function. It could undergo apoptosis, which is something called programmed cell death, or the cell gets killed off in a very neat and tidy fashion. Or it can undergo something called necrosis, whereby this is something that is referred to as accidental cell death. There's no biochemical pathway, and essentially the contents of the cell just explode. The cell explodes and the contents just leak everywhere. So one of these cellular death mechanisms is very neat and tidy, and the other one is a little bit messy. And we're going to talk about both of them. But first, some of the main ways that we actually injure our cells, some of these injurious agents, we're going to talk about the first two. The first one are things called free radicals. These are chemicals that have unpaired electrons with them. So there's a few that are present that we worry about in the body. And one of the things that you're going to notice is that the free radicals that we worry about in the body are often related to oxygen. And so free radicals that are related to oxygen, we often refer to them as reactive oxygen species, or ROS. These are produced from many different places, but two of the most common places where we actually get reactive oxygen species would be from our normal metabolic processes and from UV light exposure. We usually get rid of these in the body by antioxidants. So some of the vitamins that we intake are antioxidants, and they're going to soak up these free radicals. The more reactive oxygen species that are produced, the more likely there is that damage is going to occur, because it may exceed the ability of the body to deal with them. So usually what will happen is there'll be one of these free radicals, and there's going to be some sort of an injury to either the cellular structures or the nuclear structures or the mitochondrial structures. In particular, we're worried about the genome getting damaged by these. These three free radicals are probably the most common ones. The first one this O2 with a minus sign on it, it looks like oxygen, but it's not. This is something called a superoxide anion. The second one looks like water, but it's not, H2O2. This is hydrogen peroxide that has the ability to produce free radicals. And then this one, this OH group with a little dot beside it, this is something called a hydroxyl free radical. These are all injurious to various cellular structures. And they get rid of them by, the body gets rid of them by antioxidants. The second major injurious events that we can have is something called hypoxia. So hypoxia is a reduction in the supply of oxygen to a particular part of the body. It's the condition of having low oxygen. Oftentimes, this is going to be related to either inadequate oxygen in the air. It could be an issue with lung disease that doesn't allow us to uptake oxygen into our bloodstream, or it could be that a blood vessel is blocked off and a particular part of the body isn't getting oxygenated blood. In that case, we actually refer to that as ischemia. It's decreased oxygenated blood flow to a particular part of the body. It's called ischemia. So it's hypoxia as a result of reduction in oxygenated blood flow. If you think about what this hypoxia is going to do, well, we need oxygen to undergo aerobic cellular respiration. So the mitochondria is not going to be working very well because it's not going to undergo oxidative phosphorylation. It's not going to undergo aerobic cellular respiration. And because of that, we're going to deplete a lot of our ATP stores. Without ATP, that's our cellular currency. We can't maintain the sodium-potassium pump. The sodium levels on the inside versus the outside of the cell are all going to get out of whack. And the potassium levels are going to get out of whack. And there's going to be a massive influx and change in the amount of sodium and water that we have inside of our cell. And this is going to cause the cell to swell. It's going to cause the cell to eventually, if the agent isn't removed, those cells can die. ATP depletion is also going to cause us to undergo anaerobic respiration. And that anaerobic respiration is going to deplete our glycogen stores because you need to burn a lot more glucose in order to make the same amount of energy. Because remember, anaerobic cellular respiration only makes about 2 ATP per glucose, compared to when there's oxygen present, you're up to 30, 36 ATP. The other thing that happens is that the byproduct of anaerobic metabolism is lactic acid. And that lactic acid is going to change the intracellular pH. It's going to drop the intracellular pH, and it's going to make the cell acidic. And enzymes stop working. The cell is going to eventually die if the agent isn't removed. The third part about intracellular calcium we're not going to talk about. Just know these first two: free radical formation and hypoxia and ischemia. So depending on the amount of injury that occurs in a cell could reverse the injury. It might impair the function of the cell temporarily, but if the cell can repair itself, it does. So while the function will be impaired, it's not going to lead to cellular death. Once the agent is removed, the cell goes back to normal. There's two major patterns of injury that will occur. The first one is cellular edema. Anytime we see the word edema, it's really just the accumulation of fluid. And this is related to the fact that when you have an injury to a particular part of the body, the spaces between the cells become bigger, and the cells become more permeable. And that increased permeability causes there to be an accumulation of fluid or swelling. You can also have fatty changes, increased fat deposition. This usually indicates a more severe injury. If there's fluid it's not as severe an injury. If there's fatty changes, it is a severe injury. And this is on a cellular level, by the way, cellular level. So if we look at a reversible cellular injury, this is a normal normal epithelium of the kidney. Actually, these are kidney tubules. These are cuboidal cells, simple cuboidal cells. These are renal tubules. And this is an injured area of renal tubules. The cells still look somewhat recognizable, but one of the things that happens when a cell gets injured, even if it's a reversible one, you get these little surface blebs, these membrane blebs that occur. And you can see some of them, these little outpouches of the membrane. You can see them. There's a good one there, outpouches of the membrane. Eventually, if the agent that's causing the injury is removed, the cell can recover and go back to normal. Could go back to normal. The mechanism by which it goes back to normal is not something that we're going to chat about, but it can go back to normal. The next two types of injury are irreversible injuries. This is cell destruction through one of two mechanisms. It's either going to be apoptosis, or sometimes people say apoptosis. I say apoptosis. This is something called programmed cell death. It follows a biochemical pathway. We're going to learn the biochemical pathway of it. And the other mechanism is necrosis, which is accidental cell death. So when we talk about necrosis, this is the major pathway for cell death in a lot of injuries. If there's reduction in blood flow and you get ischemia because of decreased oxygen due to lack of blood flow, it's going to be necrosis. If there's toxins present, it's going to be necrosis. Oftentimes infections, it's going to be necrosis. Severe trauma, it's going to be necrosis. This is the major way that cells will die when they're injured. It's called accidental cell death and not programmed cell death because there's absolutely no biochemical signaling. In fact, the cellular components just simply fall apart. They fall apart, and all the stuff just leaks out. One of the things that we're going to notice about this is that, well, the inside of the cell shouldn't be exposed to the outside of the cell. But when everything leaks out, our immune cells don't recognize those things, and it causes there to be an extreme inflammatory condition. And the response is for there to be inflammation. So necrosis invariably causes inflammation. Eventually, if some of these, obviously, if these injurious agents aren't removed, the cells are going to die on a bigger level. But especially in ischemic insults, when there's lack of oxygenated blood supply, when those cells die, there's another term that we use for the tissue death as a result of a lack of oxygenated blood. And that term is infarction. So if the tissue has died because the blood supply wasn't good, we call it an infarction. While the blood supply is reduced and the cell's getting less oxygen, it's called ischemia. And then once those cells die, it's called infarction. So here's an example of necrosis. It's going from these normal renal tubules to injured renal tubules to now irreversibly injured renal tubules. And so if you get a reversible injury, you get these surface blebs. If the injurious agent isn't removed, you get a progressive injury, and the plasma membrane starts to break down, the nucleus starts to break down, the contents start to leak out of the cell, and it leads to inflammation. All of these components are going to be sensed by our immune cells, and it's going to lead to an intense inflammatory reaction. And so this would be necrosis. This is what we're seeing in part C, this third image. These are necrotic renal tubules. Necrotic, that's the term that we would use for cells that are undergoing necrosis or tissues that are undergoing necrosis. It looks necrotic. It's necrotic tissue. Necrotic tissue on a very large scale is something called gangrene. So when there's a considerable mass of tissue undergoing necrosis, called gangrene. There's two types of gangrene. There's dry gangrene and wet gangrene. Dry gangrene, you can tell because the tissue around the injured area sort of shrinks. The skin sort of wrinkles. You can see some wrinkles on the side here. And the color changes darker than the normal skin. So you can see these toes are gangrenous. We would call them gangrenous toes. There's another type of necrosis or of gangrene that is called wet gangrene. It tends to be swollen, tends to be weeping. There might be fluid coming from it. I'm not going to show you an image of that one, but this is one of the gangrenous types. This is dry gangrene on the side. The second type of injury that you can have is something called apoptosis. We're going to spend a little bit more time on apoptosis because apoptosis follows a very specific signaling mechanism, biochemical pathway. This is programmed cell death, and this is very controlled, very neat and tidy dismantling of cellular structures. Rather than necrosis that just sort of had the cell explode and leak everywhere, apoptosis does things in a much more coordinated fashion to limit the amount of inflammation, to limit the amount of damage. It will remove cellular components in really neat little packages so that none of it leaks out. Apoptosis is not always a bad thing, not always a bad thing. If there's cells of our body that are potentially cancerous, those cells are going to undergo programmed cell death, so they don't have the ability to replicate and produce tumors. So apoptosis is not always a bad thing. Apoptosis includes enzymes that degrade DNA and cytoplasmic proteins, but because the cytoplasmic proteins are degraded and some of the skeleton structures are degraded, the membrane tends to bleb off and makes these little tiny edible pieces that are completely enclosed in membrane, so that the contents of the cell don't leak out and don't elicit an inflammatory reaction. That's one of the big differences between necrosis and apoptosis. Apoptosis has no inflammatory reaction. Necrosis has an inflammatory reaction. All of these little edible bits that are breaking off from the cell that are enclosed in membrane will end up getting tagged, they'll get altered, and they'll get recognized by our phagocytes. They're going to eat those little pieces and they're going to digest it so that there's absolutely nothing that's going to leak out. Apoptosis has two major pathways to actually become activated. There's an extrinsic pathway and there's an intrinsic pathway. We're only going to talk about one of them. And we're going to talk, this is probably the most biochemistry we're going to talk about in this course, is in this section. So if you don't like biochemistry, you'll just have to put up with it for a few slides. But either way, it's not so bad. So one of the things you need to know is that apoptosis results from the activation of a very specific group of enzymes that we call a caspase. There's multiple caspases. And apoptosis, the other pieces to apoptosis that are really important in signaling for this to actually occur is a very important family of proteins called the BCL2 family of proteins. Interestingly enough, these BCL2 proteins weren't discovered until they were found to be overexpressed in B cell lymphomas. And a lymphoma is a type of immune system cancer of the actual B lymphocytes. And there's an overexpression of these BCL2 proteins in them. Because it's a family of proteins, there's multiple types of proteins under the BCL2 category, and they're separated into two major groups. There are ones that will allow for apoptosis to happen, or we call them the pro-apoptotic BCL2 proteins. And two examples of these are BAX and BAC. I don't know why they named it like that, but BAX and BAC. These are the pro-apoptotic ones. These are the bad ones. Like, they're bad. They're the ones, well, they're not always bad, but they're the ones that are going to cause apoptosis to happen. And then there are the anti-apoptotic proteins. These are still part of the BCL2 group of proteins. Now, here's where it gets confusing. In this category, one of the proteins that's under the BCL2 family of proteins is itself called BCL2. I don't know why they did that, but they did. So BCL2 is one of the proteins in the BCL2 family. And then one of the proteins is something called BCLXL. These two are anti-apoptotic. BAX and BAC are pro-apoptotic. So under normal circumstances, our cells are going to be expressing these anti-apoptotic ones, the ones that are not going to cause any cellular death. In times of injury, they're going to start expressing this BAX and BAC. So let's look at what happens in a normal cell. So this is a normal cell that has a survival signal of some sort. This cell is trying to grow, it's trying to survive. There's nothing wrong with it. So normally a growth factor is going to bind to a receptor, and this is going to stimulate the production of these anti-apoptotic proteins, BCL2, BCLXL, whatever it happens to be. And what these two anti-apoptotic proteins do is they will stabilize the outer membrane of the mitochondria. You might think, why are they stabilizing the outer membrane of the mitochondria? And one of the most important reasons for this is that within the mitochondrial intermembrane space, there are these little red proteins. Now they're red here, they're not actually red, but these little proteins that are called, it's cytochrome C. Now, cytochrome C is necessary to activate the apoptosis pathway. So by producing these anti-apoptotic proteins, you stabilize the outer membrane of the mitochondria, and it keeps all of this cytochrome C inside, and none of it leaks out. None of it actually goes off into the cytoplasm. There's no leakage of cytochrome C, and because there's no cytochrome C, you can't activate the apoptosis pathway. So apoptosis, to be activated, must have cytochrome C leave the mitochondria. This is what's going to happen in apoptosis. Now, this looks like a complicated drawing, but we're just going to be focusing on this side of it. Well, actually, we're going to be focusing on all of it. Okay. Anyways, but we're going to start here. If we just take it one step at a time. There's going to be some sort of a cellular injury. And that cellular injury is going to result in there being either no growth factor produced, or there's going to be something that's going to stop the survival signal of the cell. Whatever that injury happens to be. What ends up happening is that rather than producing those anti-apoptotic proteins, it starts to produce the pro-apoptotic proteins, BAX and BAC. And what BAX and BAC do is they go to the mitochondria, and rather than stabilize the membrane, they actually cause the membrane to become leaky. And cytochrome C leaks out into the cytoplasm. Once it's in the cytoplasm, it actually starts to activate those caspase proteins, those enzymes that are going to allow for apoptosis to happen. And there's two types of caspases that are going to be activated. Cytochrome C is going to start by activating initiator caspases. And then those initiator caspases are going to activate the main caspases that are actually going to kill the cell. And they have a very draconian name. They're called executioner caspases. And those executioner caspases are going to start to break down the nucleus, break down the cytoskeleton, and you're going to get these little tiny blebs coming off of the cell. And the cell is going to start to break off into little tiny pieces. And those little tiny pieces are eventually going to get engulfed by our phagocytes and broken down. There's no leakage because every one of these blebs is completely covered in membrane. So that's the process of apoptosis. Have a listen to it again if it was a bit fast, but those are the points that I'd want you to know about it. One of the big things that I always ask is the difference between necrosis and apoptosis. I'll usually ask you to differentiate, compare and contrast necrosis from apoptosis in 4 ways or 5 ways. And you can differentiate it by the cell size. Necrosis, it's a large cell. Compared to apoptosis, it's a smaller cell. The nucleus, don't worry about this one. I'm not going to ask you that one. The plasma membrane, we know, is disrupted in necrosis. It just falls apart. Compared to apoptosis, it's intact. It's intact. Nothing leaks out. The cellular contents in necrosis, they leak out. Cellular contents in apoptosis, they don't leak out. Everything stays intact. Because of that leaking out of the cellular contents, the inflammation is frequent in necrosis. Inflammation does not happen in apoptosis. And then for necrosis versus apoptosis, whether it's physiologic or whether it's pathologic, for necrosis, it's almost always pathologic. Invariably, it's pathologic because it's some sort of a reversible cellular injury. For apoptosis, it can actually be physiologic. You can actually remove and eliminate unwanted cells. So during our development, the webbing between our fingers goes away through apoptosis. Most of it goes away through apoptosis. And in some circumstances, it could also be pathologic. For example, if a cell has irreparable DNA damage or genetic damage, that cell is going to undergo apoptosis because that's an injury to the genetic code of the cell, and it could increase risk of cancer. So make sure that you know this chart. This chart is a really good one to know. It's really helpful to know. I think I've given you enough of a hint there. But anyways, we're going to end there. In the next presentation, we'll talk about topic 2, which will be inflammation and wound healing. Until then, if you have any questions, don't hesitate to email me or reach out, and I'm happy to answer them. Have a great day, everyone.